So here we go with the first one we're going to cover. The first one we're going to cover is called, and it's a very important receptor, G-protein coupled receptor. It's called the Beta-adrenergic receptor. So I'm going to explain the terminology here because it's very important that you understand that. The term adrenergic refers to the stimulus being adrenaline or noradrenaline, which is the same as epinephrine or norepinephrine. So the extracellular stimulus for the adrenergic receptors is epinephrine, norepinephrine. Anything that has the term adrenergic tells you that the stimulus is epinephrine or norepinephrine. The term beta is really referring to that primary sequence, that specific receptor itself. Okay? So this receptor, when it is activated, it will activate a specific G protein inside the cell. That specific G protein will activate this enzyme that I called before target enzyme or effector enzyme, adenylate cyclase. Think of the name, understand the name, adenylate cyclase. It's cyclizing adenylate. That means it's making cyclic AMP. So it's going to produce cyclic AMP. This enzyme, once activated by the proper G protein, which in turn was activated by the receptor activation, will produce cyclic AMP. So this molecule increases, goes up in the cell. Then it can go and activate another protein kinase called protein kinase A. I'll get into all the steps here in a minute, but on this page, I want you to appreciate one important thing. So I want you to sort of envision this square here as the membrane, okay? There is in that membrane another receptor called the glucagon receptor. That is also a G protein coupled receptor, but it is only activated by the peptide glucagon. Okay? So think of these two, the glucagon receptors and the beta-adrenergic receptor, as cousins, if you want. However, each one needs a different stimulus. Glucagon receptors needs a peptide. Beta-adrenergic receptor needs epinephrine or norepinephrine, the hormone. Okay? But beyond the membrane, beyond the receptor, their actions are identical. Okay? So I want you to appreciate that these are, so this doesn't work that way. Glucagon cannot activate the beta-adrenergic receptor, nor can epinephrine activate this one. So note the arrow directions, the specificity. This is to underscore specificity. The reason you have two different receptors because that may act similarly is because different tissues have different distribution of specific receptors. Some tissues may not have the glucagon receptor, but they may have the beta-adrenergic receptor. Okay. So I'm going to start with the beta-adrenergic receptor, really, which will be very essentially identical to the glucagon receptor. But let's call this entire thing here is the beta-adrenergic receptor. And here's epinephrine bound to it. Don't forget, has to be activated by the stimulus. That's extracellular, that's intracellular. the first thing that happens is you're going to have this receptor, it's going to change conformation because of the binding of epinephrine, and it is going to activate this trimeric G protein. Trimeric means it's made up of 3 subunits: alpha, beta, gamma. Note what's going to happen. You see this here, GDP. It is bound to the alpha subunit. It's going to be kicked out, coming out, kicked out, replaced by GTP. When this happens, the alpha subunit dissociates from the beta gamma. So you have the alpha subunit with GTP bound to it. Now, this is the active form of that G protein. The alpha subunit with GTP bound to it. This one is going to look for its target enzyme, adenylate cyclase. AC stands for adenylate cyclase. Adenylate cyclase takes ATP on the cytoplasmic side and cyclizes it to form cyclic AMP. So this red molecule, pink molecule here, essentially increased following receptor activation. Before receptor activation, you can assume it was 0. It was not, it was absent in the cytoplasm. After receptor activation, it increased to a measurable amount, and that qualifies to make it a second messenger. Okay? So I want you to appreciate the sequence and the identity. This is called the beta-adrenergic receptor, the stimulus epinephrine, norepinephrine. The specific G protein has this small S designation, GS alpha, with the GTP bound to it. S stands for stimulatory. Means it stimulates the enzyme adenylate cyclase, which produces the second messenger, cyclic AMP. Okay? So cyclic AMP then can do a couple of things. Well, at least one thing, which is activate a protein kinase A. And you want the A to correspond, that A corresponds to the cyclic A here. So you can, it's dependent on cyclic AMP. So cyclic AMP is a sort of an allosteric activator now, acts as an allosteric activator of pKa. And we know what protein kinases do. They phosphorylate other proteins, other enzymes, and can change their activity. Okay? So you want to know the sequence, the name, the stimulus, the G protein, the specific identity of the G protein, the active form of that G protein, the effector enzyme, and what is the second messenger produced. The only second messenger here is cyclic AMP. Okay? Now, that's basically how. Then cyclic AMP is going to activate, you don't need to know all the details here, but just think about it. Cyclic AMP binds to the entire pKa, that's the entire pKa. Cyclic AMP dependent protein kinase A, abbreviated pKa. So it dissociates the catalytic subunit from the regulatory, and then this catalytic subunit will phosphorylate enzymes and change their activity. This is what we call covalent modification of enzymes. That's right there. Adding phosphates to serine and threonine residues on enzymes changes their activity. So pKa does that to specific enzymes and can either inhibit or activate enzymes depending on the identity of the enzyme. So one important thing I want to say is anytime you activate a receptor, you want to terminate its effects. So I'm going to go back here and show you. The cell has an enzyme called cyclic AMP or cyclic nucleotide phosphodiesterase. It hydrolyzes the second messenger after a while, producing AMP. AMP has no effect. Once you remove cyclic AMP, its effects on pKa are gone. So you want to turn off that system. So that rise of cyclic A may last maybe a couple of minutes, 2, 3 minutes, 5 minutes. Then it is removed. The second messenger is removed by this enzyme, cyclic nucleotide phosphodiesterase. And I just want to show you this table. You don't need to memorize anything, but look at the list here. This is actually a pretty old table, maybe, I don't know, maybe about 15 years. But I want to just show you the point of the table is there are at least, on this table, maybe less, about 8, 10, but there's probably about 24 pathways that are affected by this G protein coupled receptor signaling pathway, by cyclic AMP and pKa. Okay? From glycogen metabolism to glycolysis to pyruvate dehydrogenase to lipid metabolism, many different metabolic pathways are regulated by cyclic AMP and pKa or the adenylate cyclase pathway. So what I want you to know is really the steps here, individual steps, the form that activates adenylate cyclase and the second messenger that's produced, and what enzyme or kinase they activate. Actually, I'm going to add one more thing. It's not on this slide. So for those of you who like to write, I'm going to tell you that other thing. This alpha subunit has enzymatic activity. And that enzymatic activity can take that GTP and hydrolyze it back to GDP. So if you replace this one with GDP, then this alpha subunit, GS alpha, is no longer able to activate adenylate cyclase. So that is called another turn-off or termination mechanism, by stopping the activation of adenylate cyclase. Because GS alpha can hydrolyze GTP back to GDP. When it's back to GDP, it reassociates with the beta gamma and it's no longer able to activate adenylate cyclase. Okay? So I'm going to say here to you that glucagon has a receptor that looks like this. Okay? Its shape in the membrane looks that way. However, it can only be activated by the hormone glucagon. It will start the same cascade as the beta-adrenergic receptor. Okay, so everything beyond the receptor between after the glucagon receptor and after the beta-adrenergic receptor will be identical. Okay? You with me? Okay. All right.